Welcome to LabNotes. I'm Joe Fetterman with Colliers Life Sciences, and today we have Vered Caplan, CEO of Orgenesis. Vered, welcome.
Hi, thank you so much for having me.
So before we start, let's just say a word about what we're gonna be talking about today. Vered has really, we were talking about it just a couple minutes ago. It's really a hero's journey about a scientist's desire to bring something to market and the challenges and the reinvention and then the reinvention again. So Vered, maybe we start at the beginning. How did you get into science anyway?
Well, I'm the type of scientist that's I'm an engineer, right? Engineers lie somewhere in the border between the scientist and kind of very practical, operational people. And I think I kind of feel comfortable in both worlds. And I was working quite a bit in biotech and doing all kinds of biotech projects. And about a decade ago, I'd actually kind of sold my former company to partners and was thinking of kind of going back more into the scientist world, right? I was thinking of going back to my academic background. And my academic background is cell engineering. It's more on kind of how do you work with biology, with cells, which are wonderful little machines, right? And I'd come across this amazing project in one of the hospitals, research institute. And to tell you the truth, when I met this product and I saw, wow, I'd come from, very kind of basic biotech, blood products, recombinants, like the really standard stuff, innovative, but standard innovative. And on the academic side, I'd always kind of looked at the future things, right? That's what you do in academy. And I was thinking, oh, now I'm going to come back and be on the science side. And when I was introduced to this project, I was like, wow, this is kind of where the two worlds meet, right? This is a practical drug development project, but it deals with cell engineering. You're using cells, and the cell itself becomes the drug. It's not a protein, it's not a chemical thing, it's a cell. And at the time, which was over a decade ago, right, maybe there were a few products in early clinical development. This was really considered science fiction. And I thought to myself, well, I'll just start this little company. It's going to be a little niche company. It's going to be like a little hobby. I'm just going to have fun with it. I'll get some grant money and some investors. I mean, this is no stress, no no big issues here. And I started working on this drug development of a cellular product, right? A genetically modified cellular product. And I could not find a good subcontractor. I found grants. I made best friends with the regulators. I got investors in. Actually, Pell Life Sciences, later bought by Danaher, were one of my initial investors. And I went out, and biotech works in a way that usually people, scientists more that are developing the drug, are not the people who make the drugs. It's a very different mindset, right? When you're innovating, you have to do something new every day. When you're doing drug production, you have to do exactly the same all the time. It's almost opposites, right? That's why if you look at the biotech industry, you see this, you've got the clever guys who are developing the drugs and the clever guys who are making them, but they're both clever in a very different way. So remember, I'd moved on to the other side. I was now on the creative drug development thing. And so I went out to get a subcontractor. A contract manufacturer would make the product, the drug that is based on cells, living cells, for the clinical trials. And I could not find one. And so I said, oh, well, what can I do? I went back to my investors and asked them, look, I need help. And as an engineer, you work with, I love engineering. I love devices. I love anything that kind of machines and stuff like that. And they told me, oh, don't worry, there is what is called cell factories. That sounds very exciting, right? A cell factory, especially, well, maybe for an engineer, it sounds exciting, maybe not for all people. So I was very excited to go see a cell factory. Now, how do you usually manipulate cells in a lab? You've got these Petri dishes, you've got biologists with this little pipette pipetting away, and you've got, they're looking under the microscope. They're almost doing like their home cooking, tasting and smelling the cells and seeing, oh, I'll add a little bit of virus and I'll add a little bit of DNA and let's see what comes out here. And that's how you work in a lab, right? But when you work in industry, it's all automated, closed system, very sophisticated robotics and stuff like that. So I was thinking, wow, a cell factory, I'm going to see something marvelous, right? So I go out and see it, and what do I see? It's a whole bunch of Petri dishes, but they're stacked. And it's not one biologist with a pipette. It's a whole bunch of them running around. And I was like, no, you can't make a GMP, good manufacturing practice. That's what you need for making drugs. I can't do this way. You've got to have something else. So I ended up working with a really lovely little lab out of Belgium, University of Brussels, and they were a little bit clever, kind of more engineering that come from GSK, from London. They had a little bit of kind of, and more mindset of how do you make, a proper GMP drug, but still very lab-based. And just when I started working with them, they didn't hardly had any revenue or anything, and I was really afraid that we're going to go. Maybe not nice to say this, but I was afraid to lose them. Because they just couldn't kind of sustain themselves. So I ended up buying them as a company, okay? And I remember going to the board and saying, we have to buy them. And they're saying, what? Why are you buying this company? They barely have $200,000 or 200,000 euro in revenue. And I said, no, but they're really special. They know how to work with this. And just after I bought them, what had happened, the first clinical results had come out of this new generation of drugs, this really fascinating, innovating world of cell and gene therapies, which is so unique, it's so different. But it's really it's almost our next stage of evolution. And when the first clinic, and these were companies like Kite and Juno, and when you saw the impact, the clinical impact of these drugs, I mean, they were taking kids, and they still do, right, that were on the verge of death. They didn't have a hope in the world, taking out the immune cells, genetically modifying, re-educating these cells, and putting those immune cells back into the patient's bodies. And it was a miracle. These kids, and if you go, I mean, you can see movies, some of these kids have already been healthy for over a decade. It's really, it's just amazing, especially when you think of cancer trials, right? Clinical trials. I mean, what's a successful clinical trials? Dying patients, we managed to have them live instead of 6 months, 9 months. Big success, right? I mean, if you think about the industry a decade ago, that's where we were. And suddenly you're having 90% remission rates, kids going home healthy. So, of course, it was wow. And everybody wanted to develop the cell and gene therapy, right? Suddenly it became, oh, we've got to do this. We're going to be the next Kite, the next Juno. There's a new, and a lot of money came in. And what happened? Happened the same thing that happened to me. Nobody could find a subcontractor. Luckily, we were around.
So you were at the right time, right place, just a little bit ahead of the wave. And,
exactly. Luck.
Yeah.
A little bit of luck you need in life, right?
Well, a little bit of luck and a little bit of problem solving, right? You were the one that you hit the problem a little bit before, and you saw that problem had to be solved. So, and this sounds exciting. What happened next?
Well, I thought, oh, okay, I've got it nailed. Now we know how to make it. And we actually set it up as a CDMO, contract development and manufacturing organization, right? So we were providing services and making these products, but still in a centralized kind of facilities, right? We were making it for CRISPR, for Servier, Iovance, Adaptimmune, a whole generation. We were working away and building clean rooms. Now, one of the things that you need to understand, why is it so challenging to make these products, okay? Why is it such a big deal? I mean, why not make it like any other biotech product? So how do you make a traditional biotech product? You have these huge stainless steel bioreactors, right? They look like these big cooking machines, right? And you pour in a whole bunch of cells and you pour in some DNA or viruses or whatever you want. You mix it all up and you harvest the protein you want, and all the rest you check out. And once you've done that, you sterilize it. What does it mean to sterilize? You kill anything that's alive, okay? And you're making these big buckets and then smaller ones, and you put them in syringes or infusion bags, and you keep them on the shelf, and when needed, you send it to the patient. That's biotech for you today. These drugs, they're totally different, right? These are living drugs. You're taking per patient, you're taking cells from the patient's body. You're doing a manipulation, but you're putting back not what the cell makes. You're putting back the actual cell. You've reprogrammed, you've re-educated that cell to go catch a cancer cell or to go make insulin, all kinds of interesting things that cells know how to do. And when you're putting back these cells and you expand them, you want a whole bunch of these cells, right? But this is done person by person, right? And the source material is the patient itself. So you're taking the cells from the patient, bringing them to a distant location, doing a manipulation, and you cannot sterilize them after. You have to do the whole process in a very sterile process, in a very sterile conditions. Because if you sterilize it, you'll kill the drug. This is a living drug, right? So you've got to do everything and that means, you know, you've got a whole bunch of biologists dressed up. They call them bunny suits, right? But you know, like you see in the people working in high contamination facilities are all dressed up. They're all, you know, packaged up, and it costs a ton of money to work this way. It's very risky because you cannot contaminate these cells. The worst contaminators, by the way, are the people working on them. So it's very expensive. Personalized, making one batch per patient, and then you have to ship it back.
Vered, I hear another innovation coming. So then what happened?
So I said, okay, this is great. We're actually making money. Imagine that, the biotech making money. And we were, you know, we were doing a lot of work. I was building clean rooms. I was, you know, technicians training them, more and more people coming out of the universities. There was nobody experienced in this. And you know what? You know, brought in private equity. They put in some money. We built out a new facilities in Houston, right? Because we wanted to serve the US companies as well. And we grew, I think, from, you know, from nothing to around $30 million in revenue in about 4 years, okay? And okay, but you know what? At that point, it was very frustrating. We were one of the biggest suppliers in Europe. How many patients could we make? I mean, how many? Maybe 100, 200 in one facility. We built another facility. Took us 3 years, okay? Yeah, now we could do 500 patients. Still, it was so, you just couldn't grow. And by the way, it wasn't only my problem. If you look at the companies in this space, it's everybody's problem, right? It's not scalable. There's no economies of scale. Taking a product, doing it in a clean room, working, you know, batch by batch, and still most of the work very manual. Even though we did put together a lot of, you know, closed systems and everything, and we were working according to GMP and doing this properly, it was still very primitive in terms of kind of industrialization, right? And at that point, you know, and this sounds like, oh, you had an economic problem. I can tell you, I wasn't sleeping at night because I had parents calling me, still do, but at least today I can give better answers, right? Calling me day after day. I had so many patients crying on the phone, asking me, where can we get these drugs? How can we get these drugs to patients? How can we do this? How can we how can we find a cure? Imagine this, you've got a kid, he's so ill, he's dying. You're reading, oh, look, there's a new cure for leukemia or lymphoma, and you can't get him the therapy. And, you know, they were calling us in desperation, but there was just no way we could make more of these products, and neither were our customers who were trying to get this through clinical trials. And, you know, it was really heartbreaking, and we were trying to find ways to work with hospitals who wanted to do clinical trials. We were trying to find ways of getting this to new locations. And that's when we had a growing realization that there really has to be a kind of paradigm shift here. So at that point, what we did, we sold the centralized facilities. We sold them with the help of the private equity. We sold them to Caplan for over $300 million, which was great, okay? And we private equity made 5 times the money. They were very happy, paid back all the debt, paid back the Belgium guy. Everybody was happy, except us, because we wanted to take that money and do some things that was really needed in this industry, not just for our customer. And the interesting thing, when you look what's happened in the last decade, remember when it started, I thought this was going to be my hobby. Well, you know, it's become my obsession. But this path, today there's thousands of these drugs in clinical development. There's been products that are approved that can barely get to patients. I mean, you can see the FDA is talking about 20 new approvals in the next few years. So it's really becoming not some niche little area of biotech. It's becoming very mainstream, because there's so many diseases that probably the only way you can cure them is by not just, you know, sticking in some chemical or protein. You really need a drug product. And what we did is we built a platform, a point of care platform. And that's kind of been the next journey. So about 3 years ago, when we sold the centralized facilities, Master Cell, that was a subsidiary, we took the money we had left and we built out what we believed is what this industry needs. And I remember I kind of promised my shareholders, my investors, look, this is going to work much faster, and it's going to be much more efficient, and it'll take me 2 years and I'll get back to the same revenue. I didn't plan on COVID, but okay. But somehow we managed, even with COVID, to get this done in 2 years. And I'm very proud of the team because they worked so hard to get this done. And now we've kind of built out a new way to make these products, make them available, make them lower cost. Products approved today, the hundreds of thousands of dollars per patient, even up to, I think the latest product was like $1.5 million per patient, $2 million. I mean, who can afford these therapies? So
let's talk really briefly about let's talk about point of care and what that means in terms of efficiency, cost savings. What does that all mean to actually, most importantly, the end user, the patient who's looking for a cure?
Yeah, so there's 2 issues here. One is cost. How do we reduce cost, and how do you make economies? How do you enable the system to have economies of scale, right? And the other issue is, how do you increase capacity quickly, right? And how do you enable these drugs to expand quickly? So the point of care platform is actually built by doing what is almost should be obvious. And really today, when people see this, they say, well, obviously this was what we needed, right? Instead of taking these cells and making them in a centralized facility and trying to adapt every time a new manufacturing modality or production modality around these cells to enable to get them to produce them and then to send them back to the patients, what we've done, we've built a system where we can actually build modular units, okay? We call them Orgenesis, mobile production units and labs. Affectionately, they're called ampules, okay? So our little ampules are about ship container size, and not by chance, because they're built into a ship's container, a shipping container. It's very easy to move them about. And we place these units wherever they're needed, right? So instead of doing the production in a centralized facility, we bring the production as close as needed to the patient. So that's one thing. And another important factor is we don't build a new production process around every new therapy. What we do is we standardize the system. We make a system, okay, that can be easily adapted to each different therapy. So you're always using the same modulars, okay, the same platform. And instead of trying to build a new production process for these drugs each time, you're adapting. What we do is we adapt the process to the platform. And we do it for customers, biotech companies, and now we can work with the hospitals directly. So we can actually provide a way to efficiently and low cost develop a good GMP, a GMP processing that is, is very safe, is very secure, but it's also very standardized. And then we can duplicate that very easily. So it's very easy to build up capacity, all right? And I think our understanding or the way we built up this platform comes from a very deep understanding of what this industry really is. We think about this as biotech, right? Oh, this is biotech. It's just another drug, but it's not. What are we doing here? We're taking cells from the body and we're reprogramming them. This is really, almost think of it as our next stage of human evolution, right? We're not stuck with the DNA that evolution threw in us. And we're not doing dangerous DNA stuff and changing, the genetic characteristic of a species. No, we're taking cells and we're reprogramming them. And cells, as I told you, are wonderful little creatures, right? They're the best little production units. They make proteins, they make a whole bunch of stuff, but they're the best little factories you can imagine. They also are extremely clever computers. They have this DNA, they have this data, right? And with the help of different technologies, you can reprogram them. You can actually, like you're using biological means, but you're reprogramming these cells. For instance, take an immune cell. You can teach it. You see this cancer cell? That's the bad guys. Now, that cancer cell was hiding in the body. The immune system was not capable of recognizing and killing it. But we take these immune cells out and we teach them. Listen, guys, you see, you go finish off these little cancer cells. And once you've taught them that, they go into the body and they eradicate the cancer. So what are we talking here about? We're talking not about manufacturing. We're talking about reprogramming or an information-based technology. And for that, you need standardized hardware, standardized software. You need to have a platform. It's an algorithm.
Barry, this is an incredible story. And I know we could go even far deeper. But I want to pause for a minute and just thank you. Thank you for sharing your journey. Not anyone can see the opportunities and the transformation that you did. And part of that had to do with your journey. And it's really a great story of how expertise advances, comes to a roadblock, looks at, okay, how do I solve? And then again. And so I have to wrap our conversation with two things. The first is, again, a great big thank you. But the second is what I know our Philadelphia audience is dying to know. When are you coming to Philadelphia?
Well, I'm just waiting for the invitation, right? No, we actually have customers in the Philadelphia area. We're really trying to figure out where we can place our first ampule there. As soon as possible, I hope.
Well, good.
And I just want to tell you, just so you know this, Philadelphia is the capital of cell and gene therapy, okay? There are no greater minds than in Philadelphia for making these innovative, life-saving drugs. And I would love just to make sure that all that amazing science becomes available to anybody that needs it, right? And I would, more than anyone else, love to do that in Philadelphia.
Well, we welcome you, Vera. Thank you very much, and we look forward to sharing your journey with our Philadelphia audience. So, thanks so much for your time today.
Thank you for having me. Thank you for showing an interest, and I really look forward to, ompulizing in Philadelphia.
Let's go make that happen. Thank you.